Today is a really interesting day. We're going to hear from first from Dr. Tanzin. Dr. Tanzin is at the National Center for Disease Control, and she's by training an epidemiologist, and she is very interested in infectious diseases. She's been there at NCDC for the last 15 years, and is interested in outbreaks and zoonoses and emerging infectious diseases. We actually met during the pandemic times. I was helping out with this INSACOG, the sequencing effort, and she was critically involved, NCDC was critically involved. So we actually attended many online meetings together and happened to connect. And she very kindly accepted our invitation. It's really an honor for us to have her here and to actually hear from people who are, at the helm, working to really address all of these issues on the ground. Thank you so much.
Thank you, Uma. First of all, I want to thank Uma and the organizers for having me here. It's not, it's not my routine job to really interact with the demographics that's here, young people like you. I usually interact with middle-aged people, old people. So it's good because, you all are the future, and you are the ones who have all the bright ideas, and you can make a difference in the future. And one bit of correction, Uma, I'm not really at the helm. I'm really in the front lines. So I'm not one of those people, the top guys who make decisions. So I'll talk about public health and role of outbreak investigations, right? So before I talk about outbreak investigations, I just wanted to give a little bit of context about what do you understand by public health. Like, we hear this word very often, public health, but what does it really encompass? So public health is the science and the art of preventing diseases, prolonging life, and promoting health. And it deals with big issues and big populations. If you really want to control diseases in the populations, you cannot be looking at individuals and patients. You have to deal with big problems that affect large populations and communities. So the focus of public health is always on communities and populations, and on interventions to reduce public health risks. It requires engagement with complex factors. So in the sense that it's not just people who are working in the hospitals curing patients. They are doing clinical work, but they are not doing public health. For us to do public health, we have to engage with different sectors. So we have to engage with the academia, we have to engage with the industry, we have to engage with the politicians, we have to engage with people who control the money, the finances of the country, we have to engage with scientists, and most of all, we have to engage with the communities that are recipients and beneficiaries of these interventions. So it's complex. And because it is so complex, there isn't one single discipline that has the wherewithal or the means to say that public health is, this particular discipline. It's multidisciplinary. So for instance, if it is waterborne diseases that is affecting a community, now the doctors are the ones who cure waterborne diseases, but they are not the ones who are responsible for the general sanitation, hygiene, and water supply in the community. So for that, we have the municipal corporation, right? So that's that sector that controls availability of clean drinking water. And within the municipal corporation also, it is the public health engineering who would know how the sanitation system is laid in the city. So in that sense, it's very complex. One has to engage with different disciplines to really make an impact in public health. And this is what I wanted to tell when I say that public health deals with problem solving in the community, but at the same time, it is very complex. It's not one discipline that has the means to solve public health problems. So having said that, now there is a public health approach to solving problems. And what is that public health approach? I have tried to deconstruct into these three areas. So first of all, we have to step back and define the right problem. They say that it's better to have an approximate answer to the right problem than having an exact answer to the wrong problem, right? So defining the problems. What are the problems that are really there in my community. For me to make a public health impact, I have to know what those problems are. If I do not have any idea about what the problems are there in my community, and if I'm randomly giving interventions, that's not going to happen. Recently, I went to a country in Southeast Asia. I will not name the country. So the problem with public health is that it's very donor driven. It's driven on people who have, the agendas are controlled by people who have the money. And oftentimes, there is scarcity of resources in public health. So you would have heard of these big names like BMGF, Gavi, who control the public health priorities at the global level. And they come with their own agendas. So this particular country that I'm talking about, there were a multitude of vertical programs in that country. And all those programs were driven by donors, and donors had their own agenda. So the government had no clue what was it that the community, the country needed in terms of priority setting. All the priority setting was being done by the donors. So that's a big problem in the public health space. The governments should know what are their priorities, what are the public health problems that they should prioritize, right? So defining the right problem, that's the first step. So that assessment comes from epidemiology. We'll talk about that later on. Then once we decide on what the right problems are, so how do we decide the right problems? You must have heard of these surveys that the government regularly conducts, right? The NFHS survey, the DLHS surveys, the NSSO does a lot of surveys. So I'm talking about in our country right now, the NSSO does a lot of surveys, and at the global level we have disease priorities project. So all these projects are there so that the policymakers get to know what the problems are in the community at the national level, at the state level, and even at the global level, right? Then the second thing is designing the right interventions. So once we know what the problems are, we have to intervene. And for intervention, I was just talking to Sir this morning about KFD. We have to have the right interventions. The interventions that are scientifically rigorous, interventions that are also affordable. Affordable technology is very important. If we have interventions that are not affordable, if we have interventions that are not, that the populations are not keen to take. Like during COVID, we saw that there was a lot of resistance to masking. Not so much in our country, but in the Western countries, America to name, they had a lot of rigidity in terms of why the government is imposing this intervention on us. So clearly that was not, probably it was the right intervention, but it was not communicated in the right way to the community, right? So designing the right intervention and at the same time communicating it to the populations. That's also very important if we want to solve public health problems. And then the third thing is about prioritizing the right decisions. So, there will be people, the NCD people would say that NCD is a big problem, finance us. The communicable diseases folks will say that communicable diseases is a big problem, finance us. But the availability of funds is always limited at the top, right? So how to prioritize, which health problems to prioritize. So prioritization is another factor that plays a role in terms of which issues get the money and which issues, and it's always about cost benefit, it's always about cost effectiveness. So if I put in N dollars in this public health problems, what benefits will the society reap in terms of prevention of disabilities, prevention of mortality, and so on and so forth. So priority setting and implementing strategies and monitoring the strategies. So in public health, epidemiology is that basic quantitative science of public health that helps decision makers provide evidence for various, in terms of what the public health problems are, in terms of what the interventions are, and how the interventions are going to work in the community. And the role of epidemiology in public health is, these are some core functions of public health that I have listed. One is surveillance. So do all of you know what surveillance means? Surveillance is systematic data collection in the community to be able to assess the disease burden. So when we looked at that circle, the first thing that I said was we have to decide what the public health problem is, right? So deciding what the public health problem is comes from surveillance, surveys, right? Then outbreak investigation. So outbreak investigation is whenever there is a public health problem, there is increase in number of cases and deaths in the community, we have to go out into the field to see what are those factors that are leading to increase in diseases and deaths. So a systematic approach of investigation towards outbreaks. We'll talk about it in the subsequent slides. Then to do epidemiological studies to assess the determinants and distribution of disease and health-related events in the community. That is also a core function of epidemiology. And then evaluation of public health interventions. So interventions have been provided, but to see whether those interventions are working or not, to course correct, is also important and is a core function of epidemiology. Then population research, clinical trials, these are all functions of epidemiology. And the first four that I have marked are related to field epidemiology. Field epidemiology is a sub-discipline of epidemiology where officials or scientists work in a field setting. All the others are either done in a research mode or done in a clinical setting. Okay, so that is, we always, as scientists, we always try to mystify things. In one sense, this is just mystification. All of this is really epidemiology, but this is field epidemiology is slightly different from clinical epidemiology and epidemiology that is used in research, because the time pressures of field epidemiology are very different from epidemiology that is done in a research setting. So that is why this new discipline was born, which was called field epidemiology. So this person whose photograph is shown in the right pane, he is considered to be the father of epidemiology. His name is John Snow. And John Snow was the one who is said to have practiced epidemiology in a systematic way while trying to solve the problem of cholera epidemic way back in the 1850s in London. So in the 1840s and 50s, there was a huge outbreak of cholera in London. And at that point in time, cholera was not associated with Vibrio cholera. The theory that was popular in that time period was that cholera was due to bad air. It was called the miasma theory. So this person, by using a systematic approach of investigation, showed that it was actually linked to drinking contaminated water. And therefore, he is attributed to be the father of epidemiology that we know in modern epidemiology that we practice today. So outbreak, now jumping from public health to epidemiology to outbreak investigations. Outbreak investigations are really considered to be an epidemiological tool for problem solving again, to understand the root cause of a problem. So problem here, when we talk about problems in the context of outbreak investigations, the problem is in terms of increase in the number of cases and deaths beyond what is expected. So the definition of an epidemic is increase in case numbers or increase in deaths or any health-related event, not just disease, beyond what is expected. So there is a statistical tool, and all of you who are from the mathematics background would know how to calculate that expected threshold, right? So investigating an outbreak is really important in the context of preventing and controlling the disease. Whenever there is a rise in case numbers in the community, the decision makers, the community leaders are always interested in knowing how to control that disease, because it leads to panic in the community. You've seen during COVID, there was so much of panic initially. The schools had closed down, hospitals were closing down, businesses were closing down, and all of that had a spiraling effect on people's mental health and also the economy at large. So many people lost their livelihood during COVID, right? So the concern of the people who are at the helm of affairs is to depress the cases, suppress the cases. So COVID was a very big example, but let me take a small example of Nipah, right? So whenever there is a Nipah outbreak in Kerala, the administration is like, really on fire. They are absolutely panicking because one, it is bringing a lot of pressure to them. It is bringing a bad name to the Kerala government. The other states sort of shut their boundaries, and people, I remember in 2019 when Nipah happened in Kerala, Kerala exports a lot of fruits to the Middle East, right? And the Middle Eastern countries had taken out this advisory saying that that the countries should stop importing fruits from Kerala. And Kerala also exports a lot of migrant workers to the Middle East. So again, there was an advisory. So those kind of things have a spiraling effect on the economics of that area and at a larger level for the country. So there's a pressure to suppress the number of cases and do it promptly, right? So to prevent and control the disease. And at the scientific level, the role of outbreak investigation is in really trying to understand what is the spectrum of illness, who are the people who are at risk, and what are the factors that are leading to increase in number of cases in terms of what are the determinants or the risk factors for that health problem or disease. To conduct research and answer scientific questions. Yesterday, all of you were having a lot of discussion about the ecology of bats and the microbiome of the bats and so on and so forth. So those research questions. And like I mentioned, political legal reasons, right? So economics and really this concern and panic in the community. So political reasons. And then we also undertake outbreak investigations to train our own staff into doing investigations.
So that's really cool to see this, that actually research and answering scientific questions is a priority as part of an outbreak investigation.
Yes.
That's really nice. That's really good to hear, I guess, for all of us.
See, so I just want to add here that research forms the fundamental of what we do in the community. If there weren't good research, then it would be very difficult for people like us to go out into the field and answer those questions quickly. Like if I didn't know that Nipah is linked to the virus being harbored by bats as reservoirs, how would public health officials and field epidemiologists such as me, would we be able to cut the chain of transmission? We wouldn't be able to do that, right? So we need research. This morning I was talking to sir, sorry, I didn't get your name, sir. Dipya Swami ji. So I was talking to him and he said that they are developing a rapid diagnostic kit. So that comes from research, right? If there is a rapid diagnostic kit, it makes our life as people who are in the business of controlling diseases so much easier. If you remember during COVID, in the beginning, there were no rapid diagnostic kits, right? And there were only very few centers in the whole of the country. NIV and NCDC were the only two centers that had the ability for diagnosing COVID. And that made just identification of cases so difficult, right? So if there weren't a group of researchers who were picking their brains to develop rapid diagnostic kit, which really changed the scenario in terms of how we approached COVID management, detection of COVID cases, we wouldn't have been able to do what we did, right? So it is very important that We do research in public health problems, because that forms the backbone and the fundamental of how we go out and do prevention and control of diseases. So outbreak investigations are, again, it's not the domain of one single discipline. It's multidisciplinary. We have our epidemiological interventions, which are really at the center, at the core of what we do, but there's also lab testing involved. If there weren't lab tests available, we wouldn't be able to detect cases, right? And there is environmental assessment. So especially in the context of One Health, environmental assessment has become extremely important. Veterinarians have become extremely important in our outbreak investigation teams. So Darshan would know, because he has worked in the KFD space in India, that every year whenever there are KFD problems in the country, NCDC or the state government, whoever sends the team, we always have an entomologist in the team, because epidemiologists don't deal with vectors. We don't understand vectors. We understand human cases. So having an entomologist becomes extremely critical to taking forward a KFD investigation. So depending on the type of investigation, we have to have people of different disciplines. So if it is a foodborne illness, we have to have people with from the regulatory side in a foodborne illness outbreak, because it is the regulators who are supposed to handle the food samples. It's not people from the human health side who test the food samples. We only test human samples, human stool samples and vomitus samples. So why outbreak investigations become important is because if we do outbreak investigations on time, and if we institute prevention and control measures on time, we are able to save ill health, diseases, and even lives, right? So if we intervene at early, we will be able to avert all these cases, right? If this is how the peak would have been if we do not have any prevention control measures. But if we institute prevention control measures on time, we suppress the cases, the case numbers. We would still not be able to control it, but we would suppress the case numbers. And that is important when we look at things from a larger public health perspective. So the usual sequence of events is that by the time the outbreak happens in the community and the surveillance system is able to generate a signal, we are at this part of the outbreak. So we are able to prevent very little. But in the previous case, the previous graph that I showed, if we pick up signals early, if we have good surveillance systems in our community, in our population, in our country, then we are able to generate early warning signals. So by the name itself, these are early warning signals. And when we identify an early warning signal, we evaluate the early warning signal, we decide whether we want to, go out into the field and intervene. And if interventions are put in time, that leads to averting diseases, deaths, disability. So again, it's in India, I forgot to show you that the IHIP system, which identifies outbreaks in the country. So till last week, there were about 300 to 400 outbreaks that were identified by the IHIP system, the Integrated Health Information Portal system that NCDC runs to identify outbreaks in the country. So, it's simply not possible for us to investigate each and every outbreak, right? So what is it that determines whether we want to go out, send people into the field to investigate, is this grid. So if the agent is not known, and if the source and mode of transmission is not known, we absolutely want to go and investigate that outbreak to be able to find answers or clues for what could be the possible agent and what are the sources and modes of transmission. Because if we know the sources and the modes of transmission, only then we will be able to institute interventions. If we don't know if we don't have a clue, if it is an unknown disease, disease X, what interventions will we be able to institute, right? We wouldn't be able to institute interventions. So if we don't know it's a person-to-person transmission, or if it is through food, through water, through contact with animals. So yes, if we don't know anything about the disease, we absolutely want to go and investigate it, and also institute some control measures. If we already know everything about the agent, and if the source and mode of transmission is known, for instance, cholera. So we know everything about cholera. Cholera has been with us for more than 100 years now, much from the middle centuries. So in those cases, we really don't want to use our resources in investigating it. We would probably want to first start with the control measures. We already know that giving safe drinking water would end the cholera epidemic, right? So in those cases, investigation takes a less of a priority, and instituting control measures straight away is more of a priority. So it's always a balancing act between what you know and what you don't know in terms of the agent, in terms of the source and mode of transmission. That is really a guiding factor for deciding whether we want to go and investigate an outbreak. So not all outbreaks are investigated, but there are some outbreaks that we absolutely have to investigate based on this scientific criteria. And there are also political reasons. Sometimes there are a huge number of deaths. We know that it is due to JE, for instance. We already know about, sorry, yeah.
One question.
Yeah.
So there are two things we are talking, outbreak investigation, of course, and then you're also saying the surveillance is also very important, which is the first layer of the things. So my question is, once we identified, say, I'll just give an example of Nipah virus. Almost always the outbreak has been reported from Kerala. Considering this question I asked.
Except one, which was reported from West Bengal in around 1990s.
I meant in the recent past.
Yeah, in the recent past, correct.
So the question I have, so there are the two questions I have. First is that the reason those are reported from one place is because you have a very good surveillance in those areas. Correct. Considering the distribution of the bats or the other thing is so widely distributed in India, that's one thing. And B, once we know like these are the places which are thing, are there measures taken to make sure this kind of being prevented? Because it's not happening every time, right? So how would you?
Very good question. So you know what, I'm sorry I didn't get your name. I didn't get you. Farah. Yeah, so Farah raises a very pertinent question about surveillance. So surveillance is that critical tool that helps us in identifying cases. How the surveillance system is implemented in our country is not uniform. So there are some states like Kerala which have very strong surveillance systems. And then there are some states which do not have strong surveillance system. So the number of cases that we detect is really a function of how well the surveillance system is performing in the community. The reason why Kerala detects Nipah year after year after year, and sometimes when we are in NCDC, we joke that Kerala is the hub of all problems, but that's not really true. The reason Kerala is detecting so many cases is because they have good surveillance system. We have to give it to them, right? And the reason why Bihar is so silent is because Bihar doesn't have good surveillance system. And that is why it's so silent. And it's the silent areas that bother us, because we don't know when that big thing would become an explosion, and then we'll be able to get to know about that. So I was talking to Uma yesterday about Northeast. So Northeast is, if you look at Northeast, it's largely silent. And that's because surveillance is pretty, it's work in progress in the Northeastern areas. The surveillance again is a function of the health system. So the health system in the Northeast areas and in parts of our country, in the central part of our country, is still work in progress. So we're working on that. So that's answering first part of your question. And what was the second question?
Second is when we know these outbreaks are happening.
The control measures, yes. So, Kerala government already has because Nipah has happened so many times, so they are a well-oiled machine. So the Kerala government knows exactly where to send, when their index of suspicion for Nipah is very high. So whenever there are encephalitis cases, they know where to send the specimens. They know what control measures to institute. So in 2019, if you remember, there were, when cases happened, that was, I think, the first time, no, 2018. That was the first time when Nipah was identified in Kerala. At that point in time, unfortunately, their infection control practices were not good. Their case detection was not good because, understandably, that was their first encounter with Nipah. So there were hospital person-to-person transmission, and there were hospital-acquired infections. And Nipah has a very high case fatality rate. So if I remember, in that cluster, there were, I think, no survivors or very few survivors, right? So in terms of how they managed the outbreak in 2019, 2018, to how they managed it last year, I think there has been a sea change. They have been able to put very good infection prevention control practices. They have a very good algorithm in terms of how to detect cases, right? So yeah, so they do have prevention and control measures in place. You had a question.
Question, but more just a comment, just comparing to Bangladesh, where surveillance has like progressively gotten better over time. So there are multiple hospital systems now that have like good case definitions for Nipah, and so when a Nipah case happens, like they can usually identify it. But the trouble is many people do not show up at hospital for them to be detected, and so that's kind of a failure of kind of any surveillance system. Is there's always unknown cases, and there was a paper a while back that actually estimated that accounting for the failure of people to show up at a hospital and also the lack of surveillance in many parts of Bangladesh, like maybe 50% of Nipah cases are undetected.
Yeah.
So I'm sure the same thing is happening here, especially in some of these other states where there is no surveillance at all.
Yes. So you raise a very important question, and this is called the iceberg phenomena. So if these were all the cases in the community, the surveillance system is really able to pick the tip of the iceberg, right? And so now the thinking is changing, especially post-COVID. The way we have started approaching infectious diseases and public health has seen a paradigm shift in terms of how we improve on our surveillance systems also. And no one surveillance system can be able to detect all the cases. So if anybody says that, they are lying. You should just say that they are lying, okay? So the buzzword these days is collaborative surveillance. So what do we mean by collaborative surveillance is that we have to look at multiple sources of surveillance. We have to look at multiple sources of data to get a handle on the situation, to be able to detect early warning signals. So there's a case-based surveillance, there's a lab surveillance, there's event-based surveillance, there's media scanning, and all those should integrate and converge. And that is what we are talking in One Health right now, right? So we are talking about surveillance from the animal sector, converging with surveillance from the human sector, converging with serosurveillance, converging with lab, genomics, and that's how we'll be improving. We are work in progress, right? Surveillance is work in progress. So this is how we'll better ourselves, we'll improve, and we'll reduce that time period from when an event has occurred in the community or at the interface between the animal and the human health sector, and the time when we are able to detect it, right? Okay, so can I move? Yeah. Okay, so talking about outbreaks, outbreaks have different patterns. So these are the two broad patterns that are described in textbooks and that we see. There are common source outbreaks and there are propagated outbreaks. So propagated outbreaks are really when it transmits from person to person. And common source outbreak can be point source, intermittent, continuous. So I'll show you an example. So if there is a gathering, a marriage, and if some food is contaminated, and if that food is eaten by a large number of people, that would be an example of common source outbreak. Usually, if this is how a typical foodborne illness outbreaks looks in our country, where it is where foodborne illnesses happen due to contamination from one single source. All the cases occur within one incubation period, and it's usually short lasting, right? So this is an example of point source outbreak. Then there are other examples of foodborne illnesses, like in countries where there are big food industries and food is supplied from one common source, large batches are distributed across the country, the outbreak would not look like this. It would look like a common source outbreak. So in our country, we don't have such good surveillance systems, so it's difficult to detect those common source outbreaks that happen in the US and Europe. But this is how a common source outbreak looks in our country. If it is contaminated water, for instance, this outbreak happened because of contaminated water systems, and it lasted for many months. You can see that it lasted for about 3-4 months. It began in the first week of January in 2024, and I think the cases are still happening because the incubation period, this was an outbreak of hepatitis and leptospirosis, and both have large incubation periods, and it was very difficult. There were multiple sources of water that were contaminated in this district, and it was difficult for the agencies to control the outbreak at one point in time.
So I do a lot of environmental surveillance for COVID here in Bangalore.
Okay.
And I think going the way things have gone during the COVID, I think this data, as we just mentioned, collaborative surveillance, I think this environmental surveillance data was very complementary to the clinical surveillance, and also considering now we don't have any testing, it really provides very good valuable information, not just for the increase in the number of the viral load, but also genomic surveillance that we do. So in real time that what variants are coming. This all can be done for hepatitis and all of these things, what we are sharing here for cholera and everything. The very fundamental question I have is why environmental surveillance has not been perceived as one of the epidemiological tool or surveillance tool, especially in the areas where we do not have any active surveillance from the humans, and especially when people are not going to the PHCs and all to get detected. I mean, this will provide valuable information, right? So do you have any insight, thoughts about that?
I agree. I concede that it provides valuable information. Too, I concede that not much resources have been invested in environmental surveillance. But now things are changing. Health sector has always been underfunded. Yeah, it's done for polio very successfully, because for polio there was, like I said, health is driven, health agendas are driven at the global level. So for polio, there was that's a different story. There's a lot of funding continuing. There's a lot of political investment in polio. But for all the other, hepatitis A is not really a problem of the Western countries. It's our problem, right? So our policymakers have to be convinced. Yeah, our policymakers have to be convinced. But as far as environmental surveillance is considered, things are changing. Now at the national level, we are talking about environmental surveillance. I never heard about environmental surveillance except for polio prior to 2020. So things are changing. Now I hear very frequently that we should do environmental surveillance, and a lot of money is being invested. Like my own institution, NCDC, has an environmental surveillance wing established, and we are trying to do sewage surveillance for respiratory pathogens. So yeah, maybe the path to change is slow, but we'll get there. And also, not many people do environmental surveillance, in India. During COVID, I know that INSACOG had a big push, So we were able to identify labs that do environmental surveillance. I'm not a microbiologist, but I'm told that it is also very challenging to be able to do, to concentrate the sewage and.
Now the protocols and everything is very streamlined. And I think this would be, maybe we can talk about it later, but this would be a good time to think about what else we can put in this, just one sample that can be used based on the priority of that area, what one want to be screening for.
Yeah, sure. We'll talk about it. Yeah, so this is an example of common source outbreak, and these were the water sources that were contaminated in this block, Agya block of Goalpara district in Assam. Then propagated source outbreak, like I said, it's person-to-person transmission, and all of us are very familiar with this COVID-19 transmission curve in India. So we had 3 waves of COVID-19 across 2 years, 3 years. And this is how a propagated source outbreak looks like. Multiple peaks with several generation periods, and outbreak is sustained because there's person-to-person transmission established. And it ends when the susceptible population is exhausted. So in our case, we had vaccination. So we had vaccination, and of course we had, this force of transmission. So everybody either got vaccinated or got infected or died. So the susceptible population sort of got exhausted. And so coming to the steps of outbreak investigation. So I am not here to teach you outbreak investigation in one lecture. It's not possible. I'm just going to open a brief window to sensitize you people about what outbreak investigation really entails, so that it can pick your curiosity and you can read about it and try to find more about it. So this is the conceptual framework of how we approach an outbreak investigation. There are essentially 13 steps, and many of these steps do not take place so sequentially. It's not so clean. They take place quite chaotically together. The latter parts take place earlier, and so on and so forth. So the first step is confirmation of an outbreak. Before we jump into an outbreak investigation, and when our surveillance system gives a trigger or generates a signal, we try to confirm whether it is really an outbreak or not. And there are several ways of doing it. The data way, I told you that we compare the observed with the expected, and if the observed shoots above the expected, then we say that this is an outbreak and we need to investigate it. Then we verify the diagnosis with clinicians, with our lab counterparts, construct a case definition, and try to identify cases systematically. And the idea is to develop a hypothesis about why this outbreak is happening, why cases are rising now, why deaths are happening now. And develop a hypothesis quickly and implement control and prevention measures quickly. So that's always this time pressure to do something in the community, right? The administrators always want to be seen as doing something. They cannot wait for the scientists to complete the outbreak investigation in 2 months and then say, okay, my scientists are completing the investigation. After 2 months, when they give me a report, I will do something. That's not how it works in the real world. So they have to do something from day one. So regardless of whether we know it's Nipah or not, if there are people who are dying with brain deaths, acute encephalitis is happening, regardless of whether the scientists have confirmed or not, we have to perceive, perception is everything. So we have to make it look like we are in charge and we know what we are doing, right? So implement prevention and control measures immediately. So this is how, like I said, there's descriptive analysis of data, and then do analytics to test our hypothesis and then response, but it does not happen so sequentially and cleanly.
Just a quick question. So it's very much like a kind of a crisis thing.
Yeah, it's a crisis,
so,
always.
Just to give a perspective, like as a scientist, say, you have the same kind of job every day, sort of. I mean, of course, it changes what you do every day. There's no urgency as such. There is urgency sometimes, some students have to publish or you have to submit something for a grant proposal, something like that. But this is a very different kind of thing, right? You have to, like it's a response. It's like, I don't know, being in the fire department or something.
Yes.
You know,
In
where
the
you have
line
to
of fire.
immediately go and kind of on call.
Yes. And also, I want to add that there's one thing that there's always an emergency. Then the second thing is that, there's this, you never know when there would be an outbreak, right? Like you would know when your thesis submission time is, or your research grant time, or whatever other things that you have, but we never know when an outbreak will happen. So it's always like, and it always happens on a Friday. I've been in NCDC for 15 years. And when we are just like, we're like, okay, end of the week, we are going to wind up, and they will say, there is an outbreak in, district so and you have to go. And you're like, oh my God, why a Friday? So yeah, so there's that thing. Yes.
I hope I'm not jumping the gun here, but is there a standard operating procedure that you have in case any outbreak happens? These are the set of things that you do, no matter what the pathogen is. You don't know what it is. Is there any SOP
So,
for it?
I wish there was such an SOP. But no, there isn't any such SOP. But broadly, if it is waterborne diseases, you would just say, okay, boil the water. Or if it is a virus that's not affected by chlorine, you say just distribute chlorine tablets, or chlorinate the water, super chlorinate the water, boil the water. So those kind of broad guidances are there. But trust me, like this is from experience, every outbreak is different. Every outbreak is different. Even if I have investigated 10 outbreaks of cholera, the 11th outbreak would be different from all the 10 others that I have investigated, because the setting is different. The people, the risk groups are different. The administrators are different. So state to state, it varies. If I go to Assam, Assam's response is going to be very different from Kerala's response. Kerala's response is going to be very different from Maharashtra's response. And so that is another challenge of field epidemiology, is that we are working in the real world, in a real world setting. So real world settings are very different from our own controlled academic environment.
So in fact, because also that health is a state issue,
Yeah.
right? So there are maybe, it's the same as wildlife. So wildlife also is regulated by the state. Of course, there are national laws and there is a MOEFCC ministry. But for example, to work in Assam, I need to get permits in Assam. I need to work with the forest officers there, those kind of things. So
Absolutely.
you need to collaborate, but depend on them also in some sense.
Absolutely. Many times, there are so many times when there have been outbreaks of, say, cholera in Odisha, and we are at the national level, because we have the IHIP, we get to see everything. We know that this outbreak is spreading from district to district, and when we call up the state and say, hey, can we investigate it? They say no. So a lot of reasons. And also it depends on who is where, like, if politically, if there is one party at the center, and if there is another party at the state, and those two parties don't see eye to eye, then they don't collaborate with us. And sometimes, there are data sharing issues, many things. So anyways, so regardless of all that, we continue to do what we do. So this is an example of descriptive epidemiology. So why descriptive epidemiology is in the context of why do we first want to understand and do descriptive epidemiology, is to define who are these people who are at risk and who are getting the disease. So this was an outbreak that we investigated in Uttar Pradesh. It was a simple diarrhea outbreak. So you can see that these were mostly children, and the attack rate was not uniform across the village. There was an area where the attack rate was very high in comparison to the other areas. So this is how it looked like when we plotted it. And so when we saw that area A had a higher attack rate in terms of in comparison to the other areas, we really were able to zero in on the water sources, and we could prioritize the intervention in these water sources. So in that sense, descriptive epidemiology is important. Then the mpox cases. So this is an example of mpox cases in India. So, we heard about mpox outbreaks in the West, in Europe, and we saw that, it was spreading in the general community, and it were people with a certain, people who were MSMs who were vulnerable to mpox in Europe. But when we looked at in our own country, the few cases that we were able to detect, we saw that it was not in the general community. It was only amongst segments of people who were expats, either expats in India or expats working outside of India but had returned to India. Okay, so this is what the data was showing. And so similarly, we didn't see any sort of connection with, good morning, sir. So we didn't see any connection with, MSM sexual orientation as far as mpox was considered in our country. It was really all the people who were affected had heterosexual orientation. So the intervention, the reason why I'm showing you these slides is because with this information, our intervention, our prioritization on the risk groups would change, right? So this local information is very important in terms of who we prioritize for intervention. So this is an example of time distribution of cases, what we call an epi curve. So here, this is not a point source outbreak. This was a diarrhea outbreak that I showed earlier. You can see that it's really like a common source outbreak over, I think, several weeks, spread over several weeks. And so when all the information from descriptive epidemiology comes together, we put them all together and we really evaluate the information and try to generate a hypothesis in terms of what could be the possible pathogen, what could be the source, and what would be the mode of transmission. So if it's a short acting, like a point source outbreak, we know that it's a pathogen which has very short incubation period, spread over hours or days. But if it is common source, then we know that it's possibly something with a longer incubation period, a different mode of transmission, a different source. Okay, and then we develop an hypothesis on who are the people who are at risk of becoming ill, what would be the source and the vehicle, and what is the mode of transmission. And these are some examples of typical hypotheses that are developed during an outbreak investigation. So for instance, tattooing is a risk of getting hepatitis B infection because 13 out of 15 cases had new tattoos. So usually in outbreak investigations, we don't deal with large sample sizes. These aren't population studies. We deal with very small samples. And that is why we are not able, oftentimes we are not able to publish it because, when journals look at our articles, they say, oh, it doesn't have enough power. But how do you control the power? How do you control the number of people who are getting infected, right? You can't. It's a natural experiment. So another example, a shallow well was the source of Shigella because most of the cases used water from there. A juice from the school cafeteria caused the illness because a pass-by visitor got sick after drinking a glass of juice. So outliers are also very important when we are generating a hypothesis. In fact, if I take you back to the story of John Snow, so John Snow was able to link all the cases of cholera to the Broad Street water pump, the tube well pump, and he saw that there were some cases. There was a local brewery. So people who were working in that brewery were not infected by, they did not get cholera. And when he tried to get more information, he realized that actually those people were not drinking water. They were happily drinking only beer, right? So those were outliers, and that kind of gave him a hint about linking the cases to that water pump in Broad Street. So we always, when we are training our officers, we say, look at the outlier cases. So lab studies and environmental studies. So lab studies are also very important. This is an outbreak that we investigated in Andhra Pradesh following very heavy flooding in Eluru district. In Eluru, Western Godavari district. And because of the flooding and heavy pesticide use in those areas, there was pesticide leaching somehow in their water source, in the municipal water source. And so, while we were epidemiologically able to demonstrate that drinking municipal water was a risk for acquisition of this acute encephalitis, neurological illness, but that really didn't clinch till we got an environmental sampling done by our colleagues from ICMR colleagues in IOH, and they found that the pesticide levels, all sorts of pesticide, were way above the acceptable limits. So it's not, what I'm trying to emphasize here is that it's not just the epidemiological study that clinched the reason for this outbreak. It was also the environmental study, the limits of the pesticide that was found by colleagues who work in the environment domain that clinched the reason for why this outbreak happened. So that's the descriptive part. So we do the descriptive study to enumerate. We do a descriptive study to generate a hypothesis, and then to institute prevention and control measures on the basis of who are the people at risk, and what could be the possible risk factors for or determinants for the disease or ill health. Then the analytical study. So it's just like any analytical study that you all would be doing. So in the descriptive study, there is no comparison group, right? We are only looking at the cases. So to generate the hypothesis. But for the analytical study, where we want to prove or disprove our hypothesis, we have to have a comparison group. We have to have a group that does not have either the disease, if we are doing a case control study, or that does not have the exposure, right? So we can go either from exposure to disease or do a retrospective cohort study. Usually we never do a prospective cohort study, we do a retrospective cohort study. So this is an example of a retrospective cohort study where we looked at those who ate food at a particular food item versus those who did not eat the particular food item, and compared the proportion of cases in terms of those who ate with those who did not eat, and calculated the relative risk. So a person who ate fermented vegetable was 6 times more likely to be ill than a person who did not eat. So this fermented vegetable was considered as the incriminating agent for this foodborne illness. Then a case control study. In a case control study, we go retrospectively. We compare the cases to the non-cases, and we look at the exposure status. So here we calculate an odds ratio. So in this foodborne outbreak, eating kheer was suspected to be the implicated food, as a person who ate kheer was 200 times more likely than an ill person who did not eat kheer. So this is another recent example of a KFD outbreak that we investigated. Here we found that we did a case control study, and we found that people who cited recent monkey death within 500 meters of their house had a higher odds of illness, and those who had, of course, household exposure to ticks. So this is a well-known fact, a well-established fact, but we were able to demonstrate this again in this outbreak that we investigated. And like you can see, the number of cases is really low. To increase the power, we try to take more controls, but again, it's always a challenge for us to have a good analytical study with sufficient power. So trying to conclude now. So, John Snow tried to control the cholera epidemic way back in the 1850s and 60s by removing the handle of the water pump. So he did that descriptive study where he was able to link all the cases to this Broad Street pump, and the intervention that he proposed at that point in time is remove the handle of the pump. People will not be able to draw water, and voila, the number of cases dropped, right? So the last outbreak of cholera happened in 1866, and John Snow's observations led to a major breakthrough in understanding cholera. So a person with an astute observation, right, and he was able to say that, no, it's not bad air, but it's actually drinking water from this water pump that is leading to illness, way before the germ theory was postulated. People didn't know that, germs existed. So in that sense, this was very path-breaking. This is a person who was ahead of his times, and he just looked at data. That's all that he did. He looked at data, and he made that temporal, he made that connection with this water source. So that is the power of observational studies and data. And using that analogy of removing the handle of the water pump as an intervention, all outbreak investigations are done with this singular thing at the center of our thought, is that we have to have evidence-based interventions. All outbreak investigations are done to generate evidence for improving public health practices and for instituting evidence-based interventions. So I said in the beginning, in terms of what the etiology of the disease is, in terms of what could be the source and what could be the potential modes of transmission. So these are some interventions that we routinely institute. I think somebody asked a question to me about, are there any standard templates? So there aren't any standard templates, but mostly the interventions that we institute are guided by these broad principles. We have to institute intervention to interrupt the transmission chain. Okay, so how do we interrupt the transmission chain? We remove the contamination source. So if it is a person-to-person transmission, we would give a recommendation for masking, drinking non-contaminated water. In case of Ebola, you know that contaminated dead bodies became a source of contamination, because people had, in Africa, people had this practice of washing dead bodies. We have that even in our country. So even during Nipah, this was one intervention that was recommended, that burials have to be done in a certain way, that not many people should congregate for burials, and washing of the corpse should not be undertaken, right? So safe burial practices. Then remove person. So this is another thing that the Kerala government does, whenever there's a Nipah outbreak, they take out some SOPs, and one of the components of the SOP is safe burial practices. Yes.
Question. So this is a couple of slides back when you were talking about the cholera outbreaks, and you said the data, the sample size is so small that some of the time it's very difficult to publish. The thing I have learned in public health is you have to make things really inform the government first before anybody thinks about the publication aspect, which is very true, and I think that's why we all do what we do. But my question is that even if these are small number of cases and they're happening every now and then, these data sets are very important, actually.
Absolutely.
So the question I have, that is this kind of information available somewhere? So it may not, now the outbreak has happened, at mitigation has taken place, but over a period of time, this can be very useful in order for someone else to look at them, plot them, to understand, like for example, NASA does these very nice GIS and all kind of laying, where are the flooding risks, where the cholera outbreaks can happen. So it may not be the priority of, say, any department where you are. Maybe someone else might be interested in looking at and say, look at the district level or at the state level, these are the hot spots which are likely to happen at this time of year or something. So where is all these outbreak data is sitting? That's my main
IDSP,
question.
right?
Yeah.
So the data is sitting in IDSP, but I think what you're saying, what you're asking is, what are the investigation reports? You're talking about the findings of investigations.
Yes.
So yes, very good question. Unfortunately, we don't have a culture of publication for people like me who practice public health. It's not, there are no incentives, there are no disincentives also. But out of our own sheer interest and the way we were trained, when I was trained, I had a mentor, my professor, his name was Rajesh Kumar. He's still there. I mean, he's not practicing. So he always used to say that, if you don't publish, because I was trained in a research kind of a setting, he used to say that you're not doing your job fully. So we try to publish as much as possible, as humanly possible. But to answer your question, we do have bulletins that come out that do not go through the scientific rigor of what a publication probably requires. So NCDC has NCDC newsletter. We also have an IDSP bulletin. I think it comes out fortnightly. Our newsletter comes out 3 monthly. And we also have something called a CD alert that's disease specific, that comes out occasionally whenever there is a public health issue in the country. So we take out those CD alerts. CD stands for communicable disease alerts. So those are some publications that at the institute level we try to take out. Similarly, all other public health organizations in the world have their own bulletins. Like CDC has something called an MMWR bulletin, which is very widely read. It has a very good impact factor. So the MMWR bulletin publishes findings from outbreaks that primarily it caters to the US, their own domestic environment. So during COVID, I remember, when we were grappling with the issue of person-to-person spread, but like recommendations for whether we should mask indoors, outdoors, they took out a publication which showed that singing in a choir singing was leading to transmission. So that was very helpful for us, to devise to modulate our recommendations, because what was true in US at that point in time was true everywhere. So we did take out recommendations for even indoor masking, because at that time there was a lot of debate about what should be the masking recommendations and so on. So yes, these kind of publications are important. And another publication that is also very high impact factor taken out by EuroCDC is called Eurosurveillance. So you can go through Eurosurveillance, and in India you can go through the NCDC newsletter, the CD alert, and the IDSP fortnightly bulletin, where we publish these kind of investigations.
As you said, if you want to get any data from the bulletins as you published so far, bulletins, so you have just analyze the data from your aspect. So if I want to get a different aspect I want to approach, I don't have the raw sources actually. Sometimes if you are like incidence cases, you are reported state-wise, sometimes for the nation-wise. But if I want to go a little deeper, I don't get any sources. Like I can say the National Vector-Borne Disease Control Program. I can say there is no data regarding the vector-borne diseases like chikungunya, dengue until 24. Now we are in the July 24, but I could see the data until 23 September. So I could not get any of the raw data. So it makes very difficult to collection of the data first. Then only I can think about the analysis. Like you are talking about developing the hypothesis. If I come with some hypothesis, I'm failed to get the data first of all. So this is the biggest issue I felt to work with the public health data. So if you're suggesting something to how to get the data, even if you're approaching some the government like agencies, we are not getting the response from that.
So getting public health data is always a challenge, and it is not just a challenge for you, it is also a challenge for me, right? So no, I am absolutely being honest. Like you give the example of vector-borne diseases, and because I work in NCDC, and NCDC is a different entity from NVBDCP. If I have to get the number of malaria cases in the country, I can get the number of malaria cases, but if I want a disaggregated, really granular data in terms of sub-district level incidence, trust me, it's going to be difficult for me also. So what I would suggest, as a way forward, I don't have like the right answer to it, but what is happening now is that data is getting consolidated. Right now, the data was very, very, very fragmented. So at the national level, the people sitting at the national level also do not have disaggregated data. But now I'm seeing in IDSP a change is happening. We have this integrated health information portal. The government has mandated that all the data from different sources should be put into one portal, the IHIP portal. Many programs are now coming onto the IHIP platform, and hopefully the different systems would start talking to each other. Like, for INSACOG, it was a big fight for us, but ultimately we were able to get all the data together, right? The surveillance data as well as the genomic data, you have this IBDC where all the data is stored. So it's happening. It would, right now, the data is still very fragmented. But hopefully in times to come, we will be able to have one single source where all the data is available, different operating systems will talk to each other, and it would be easier for people like you, researchers, to get the data. Right now, I know it's difficult, but maybe to answer your question, maybe you can approach the state government, because the state government is really the responsible for generating data for their own state. So if you want data for, say, Maharashtra or whichever state you're interested, you can approach the state government or the municipality, whichever level of data that you require.
Excuse me, just to add one point for this outbreak investigation related publications. Madam is also coordinator for EIS course. Since I think this course is there since last 10 years, right, in India, with the NIE, Chennai and NCDC, even it's going to be started in IISc Bengaluru shortly. So every batch, every candidate who is finishing his course, they'll be coming up with one such publication where they have been given an practical outbreak experience, and they will be sent to do an investigation. So even this data which is coming out of these courses will also guide us. And with respect to data, I'm one person where I was being a researcher who was blessed to be, my name is Darshan. Since last 6 years, I'm working for One Health related research activities. I was with the state health department working for Castle Forest disease. And with respect to data, I would like to say some instances wherein even some of the ICMR institutes had very difficult time to get data with the state. Till date, it is not resolved. So with the initiation of National One Health Mission, which is going to be, launched in 1 or 2 months, so I think this is going to be resolved. Till now, even NCDC, when they come down to the state, ma'am was highlighting, if the states don't want to cooperate, they will make sure that nothing is given out. Yeah, it's a practicality.
So just like to say, as these systems are developing, it would be really nice to be cognizant of wildlife as well. For example, when we work with rodents, we work with bats, we find some sequence data, it's just, of course, it's just co-occurring correlation, but there's some place we can put that also. Otherwise, it just goes out when it's published 3 years later, it goes on to GenBank, but we would love to be able to contribute that data in a real-time way. Basically, if you just, of course, databases are just places you dump stuff. There has to be somebody looking around to see, oh, this has come in, this is, someone who's like got a bird's eye view and is able to pull things together. We're all working, whether we're researchers or, outbreak investigation, or we're all interested in the bigger picture. So it would be very nice to be cognizant of, right? Because right now, there's absolutely no place. Everyone says, oh, you should report it. But to whom? What? We have no idea.
Yeah. So I think broadly, just to summarize this discussion, we are work in progress. In India, we're only 75 years old, right, as a modern nation. So data sharing and all these concepts are very egalitarian concepts. And right now, all of us have this tribal tendencies, right? Little bit. All of us have that innate tribal tendencies. This is mine, and I should be the owner of this. So the society, as societies evolve, systems would also evolve, I guess. So what do you think? This is a... It belongs to everybody, yes, yes. So that, yeah, you have to have that broad egalitarian thinking.
Yeah, I entirely agree with what you said, and it is with this objective that IHIP has come up.
Yes, yes.
In my talk, I'll be showing you a document of Niti Aayog, where they have recommended, Niti Aayog is going to implement it soon, that there will be a central portal where all the data will be available and you can access it through a secured login and giving a reason why you want the data and so on. So I think, as you said, we are progressing. From pen paper when I started 45 years back to now
Digital.
computer databases and so
So
on.
IHIP is really, a state of the art. Unfortunately, I somehow forgot to put that here in my presentation, but if you look at the IHIP interface, it's really state of art. And, even the most developed countries like US and all, we are at par when it comes to IHIP.
This has been a fascinating discussion. Before we finish, I'd also wanted to get your personal insights. For example, how has outbreak investigation changed, and what is changing? We know that there's been population size has changed tremendously. Inequities. There's a lot of differences in India in different parts of the country. So what is a bird's eye view of, from your 15 years of experience, if you could say, how are things changing? Are there more and more outbreaks? Are there much less? Are we getting better at investigation? Just some.
So I think, yes, we are getting better at investigation. Earlier, even before I joined NCDC, when I talked to people who have worked in NCDC for 30 years, 40 years, my seniors, the quality of the outbreak investigations that we do has changed. Earlier it would be every waterborne disease outbreak would be investigated by national teams. Every hepatitis A outbreak would be investigated by national teams. Now we do these outbreak investigations, but these are mostly done for our own, ticking our own agendas for training purposes. But the state governments have become extremely self-reliant. They don't call NCDC for every outbreak investigation. They would perhaps call NCDC for a Nipah outbreak or some unknown disease that they are not able to handle. But for every outbreak investigation, they don't dial NCDC. So that's one thing. Two, outbreak investigations have become more systematic. Over the years, we have become better at how we approach outbreak investigations. So our outbreak investigations have become more slicker. They've become more systematic. We've started using more technology. So we do outbreak investigations, when we do these epidemiological investigations, we always have to rely on data sources that are collected by others. So that's another challenge of doing any epidemiological investigation in the field. It's simply not possible for us to collect neat data sets the way you people do, because you have your own project or thesis, you collect your own data, you're in charge of your own data sets, right? We are never in charge of our own data sets. We are always reliant on other data sources. Data, for instance, collected by hospitals. And trust me, those data sources are not the best or not the neatest. And they're not collected for the purpose that, one day a team from NCDC will come and they would try to develop this hypothesis. They are collected for their HMIS, Health Management and Information System. They're not intended for our hypothesis generation or proving, disproving our hypothesis. But over the years, even now there's a revolution in terms of how the data is collected. So hospitals have electronic health records, many hospitals. It's work in progress. We have something called Abha ID. I don't know if all of you have heard about Abha ID. Raise your hands if you have. Okay, only 2 people. So there's something called an Abha ID with PMASBY, Pradhan Mantri, this insurance scheme. Yeah, the Swasth Bima Yojana. So every beneficiary is given an ID. Just like in the US and probably in NHS, they have a health ID. So a health ID is being given to every person for purpose of insurance. But what that is doing slowly is that all our health records are getting digitized. So it's much more neat, right? Any investigation I can get at click of a hand. Any number of OPD visits I can get at the click of a hand. Otherwise, we would have to sit in the MRD for days going through that mold and the dust of registers. So that has changed. So the way we collect data, and also the way we approach our investigations. Of course, our maps have become much more slick. We have GIS. We don't have to do hand-drawn maps. So yeah, things have changed. A lot of technology has come in. But at the same time, I would sound a word of caution. Technology is not it's not a replacement for the gruel of field work. The field work has to be, should not change. We cannot say that, okay, I'll get all the information from wherever, draw it from the electronic health records. If I don't consult with the doctor, the physician, the people who are actually looking at the cases, I will not be able to do a good investigation. I will miss out on something pertinent, which will then have an effect on how I generate my hypothesis, how I test my hypothesis, and what interventions that I recommend. So technology is something that helps us, but it's not a replacement for the field work that we are supposed to do in the field. Talk to people, just basic things, one-on-one interaction. Sorry, yeah. So can I move?
Yeah, sure.
Yes. Yeah, so, interrupting transmission, so removing persons from exposure. So infection prevention and control is very important. We saw that in COVID, the elderly who were in the care homes. We have to have good IPC practices, otherwise the infection would spread rapidly. Isolate and early treatment of infected persons, that's simple. Again, we saw that in COVID. The whole purpose of early identification of cases was so that we would isolate the cases and the cases would not then continue to propagate the outbreak in the community. Then modify host responses. Vaccination, again, COVID was a big teacher, a big teacher. How much we learned from COVID is another thing, but it showed all the aspects, the entire spectrum of disease pathology and how we approach disease in terms of management. So vaccination, when we started vaccinating people in the community and modulating the immunity level at the community, our case numbers came down, right? So mass vaccination. Chemoprophylaxis. A couple of years back when there were floods in Kerala, it led to a large leptospirosis outbreak and we did mass chemoprophylaxis with doxy and that led to a decline in cases. So mass chemoprophylaxis. Medicated bed nets. We use this for malaria in our northeastern part of the country and it has brought the case numbers down from several lakhs to 1.1 million cases this year. And application of repellents for KFD, that's what, vaccination doesn't work. We don't have a good vaccine. So that's all that we are left with because we cannot tell these people not to go out into the jungles. Their livelihood is dependent on going into the jungles, right? So application of repellents, use of protective clothing. So these are some interventions. Yeah, so this we've already discussed, what are the challenges that we face when we do field epidemiology, unexpected event, need to investigate quickly, pressure for answers, multiple agencies, media spotlight, and work carried out in the field. But despite all that, we have to have a systematic approach and we have to have an approach that is quick and clean, that balances good science with astute judgment. And this is just, so Darshan would know this. This is Bhavesh. Bhavesh investigated, Bhavesh is one of our officers in NCDC, and I had mentored him for this investigation of KFD. So he very kindly gave me his diary notes. He writes a diary. So just to bring you, challenges of a field epidemiologist, field work, of course, but at the same time, you don't have a template. You asked a very good question, do we have a template for approaching our investigations? We have to constantly learn, modify our scientific approaches. Sometimes we do things right, sometimes we don't do things right, because in this case we did a case control study. We tried to recruit the number of controls because case numbers were limited. This area is a very difficult area to work in if you've been to the Western Ghats. So cases, the hamlets are very sparsely distributed. So we recruited controls from the village of the cases, but in the process, when we did our analysis, we realized that we had overmatched, and our outcomes were, they were very small. We couldn't get a sufficient level of significance. So we had, this is something that he's written in his diary. So to summarize, public health is multidisciplinary. It is a problem solving, we do problem solving in public health, but it is complex, it is multidisciplinary. But at the same time, it's extremely important because using public health, we improve the health and lives of large number of people. Epidemiology is the quantitative evidence generating branch of public health, and investigating outbreaks is we use epidemiological principles, and it provides opportunities for identifying evidence-based interventions to improve practices in the community for preventing health and disease. Thank you.
Thank you so much. Any quick questions?
That was a wonderful talk. Thanks a lot. I mean, outbreak investigations and things is generally something that we never spoke about properly here, and really glad. I just have two questions. One is that, so do you also have any field or like ecologists who work with you through NCDC, or do you have like consultant ecologists in case if the diseases are zoonotic? And
so, we have veterinarians as consultants in our agency, but we do not have ecologists. But because NCDC is a national agency, we work very closely with many of these One Health government agencies. So we collaborate with different One Health agencies like Nivedi is there, and many other agencies. So we work with the Animal Health Commissioner. We work through the government.
Next is that I was just curious to know, is there, so the repellents that you mentioned for ticks against KFD, do you know what repellent this is? Because it's kind of very useful for people who work in the field as well.
Darshan would probably answer that better.
Some issues with the quality of the liquid which was supplied in the field. So the recent one is this was developed by VCRC, and it was licensed with DRDO. So the same thing which is being used in the border areas by the soldiers is being brought to the state, and it's working well, and acceptance is very good, because we had a field study because of the DMP department used to supply. Every house used to have these 2 bottles. As soon as you enter their house, you could see somewhere near the window, but none of them was opened. And most of them was using it for the cattles, because it had some that stickiness, that smell, odor. So what we thought is, with the field experience, again, when we had an interview, key persons, those things. So what we thought was, we wanted to make it more appealing. So we gave it a cosmetic touch. We started telling that it has, actually we also added vitamin E, glycerin, and some sort of fragrance. So now it has become, people are demanding it. So, and once they knew that, I don't know how many of you know this, every repellent which works for mosquito will work for tick, otherwise. So if this thing is known by the people at the community level, so they will start demanding. Yeah.
Thank you.
It works, but then these people go out to the jungle to collect
Yeah.
firewood, right? So it's
So it also depends on
and they're-
humidity, right? I mean,
yeah,
we know
and they're
because
also very poor.
yeah.
So this is available.
Yeah.
This is available through the government system.
So just to kind of reiterate a point that Arjun brought up, while animal health and veterinarians are, cognizant about animal physiology, I think ecologists can bring in a different perspective, which is thinking about interactions between, ticks and animals and things like that. So it will be really interesting as this One Health approach comes up, because it does have a strong element of ecology. Unfortunately, ecologists in India itself are not that many. There's not that, it's not a very big community, whether in institutions or otherwise. Mostly they're working in the NGO sector. So that's often a barrier for participation in some of these things. So it'll be very interesting to see how it comes up as a culture of collaboration.
Yeah, and it's also a learning, it's two-way learning, right? Yesterday when I was here and I heard some of those talks, discussions that you people were having, and that really, I mean, we had never approached, Nipah has been happening so many years, but, I never thought about bat microbiome till I heard yesterday, and I didn't know that, there are people who are interested in bat microbiome. So it's a learning for us also. It's not like we don't want to collaborate. Sometimes we simply don't know.
That ecologist perspective, but also when you just quickly mentioned NIV and Nivedi and all these organizations. I think the reason we handled COVID so well, because everybody was involved. There were so many labs, people were testing.
Yeah.
I think we got to also change our approach and involve. There are so many people interested.
Absolutely.
They want to. So I think that will be really nice if other labs, non-governmental or private ones, are involved in these. I saw an article yesterday, for example, in the newspaper that NIV is inundated with mosquito samples or human samples for Zika testing, and they don't have bandwidth to do that. Had that been other labs involved, I think it would be much quicker process to understand what's going on. Basically.
Absolutely. I'm on the same page as you are. And Uma would tell you, like during COVID, with the INSACOG, change takes time, but with the INSACOG, we were able to go from 10 labs to now, I think 60 labs. And there are many, of those 60 labs, there are many private labs. So it took us, we used to sit every day for 1 hour, 2 hour, rigorously debating, fighting. So yeah, it happened. But
goal for this workshop also, because we have modelers, I mean, mathematical modelers, we have molecular biologists, people who are working in NGOs, One Health Trust, Karnataka government, I mean, all kinds of people. So the hope is that, we build a culture from, the younger generation to kind of work together and, some interdisciplinarity.
Yeah, and hopefully Darshan is at a very, he's at a very strategic place right now. So I think Darshan,
if you
all our
have,
expectations are on you.
So right
If
now
you
I'm
can break the
representing
silos.
National One Health Mission, which is launched, already approved by cabinet, has to be officially launched in some event or something. That's planning is going on. If you have some time, I can brief later on after the next session, after the post-lunch session, so that most of the questions what you are asking to involve ecologists, other things, is already in the pipeline. Shortly, we'll also receive some invitations for some of the workshops. So you will get to know about it. But right now, officially, I can't announce it.
Conversations should keep going. Yes. Because if we are talking, we are getting to know each other. Like ecology is important. Yeah, that builds trust, and that's how during emergencies you can forge collaborations. Okay, thank you so much.